Over the past 7 years, Adam and Situ Studio have engaged in a long and evolving set of conversations about space, mapping, and visualization. And it's really through these overlapping set of interests that we've engaged in a series of collaborations which have really pushed us to kind of the perimeters of our respective disciplines of geology and architecture. Tonight, we're going to show you the most recent of these projects.
So in the mid-19th century, Charles Darwin was presented with a problem. The theory of evolution, the origin of species, was supremely successful. However, at the time, the oldest known animal was a trilobite, a large, complex organism with eyes, legs, tentacles, a tail, a head. Where were its ancestors? What were the earlier, simpler forms of animal? Well, at the time, his best explanation was perhaps the geologic record is incomplete, or perhaps there were discoveries yet to be made. And at the time, this was not a satisfactory answer. But it turned out he was exactly right. Since then, we found ancient bacteria, early algae, simple eukaryotes like this fungi, and even earlier ancestors of animals like this mollusk. But there remained a large gap. And interestingly, that gap was filled by glaciations, two of the most severe ice ages in Earth history, frequently dubbed the Snowball Earth. So we actually spent the last 6 years in South Australia trying to understand why there were these ice ages and what effect they might have had on life. But what we found was completely unexpected. Hosted in a bacterial reef just beneath the glacial deposits were a series of shapes repeated over and over and over again. Rings, wishbones, anvils, and perforated blobs. And we couldn't help asking ourselves the question, could these be random two-dimensional slices through some larger, more complex three-dimensional object? But at the time, we didn't have a means of extracting these forms from the rocks themselves.
So normally, X-ray-based CT technology would be used to image embedded objects. So you can see here a CT scan of the human brain, where you can clearly differentiate brain matter from bone, tissue, et cetera. This technology relies exclusively on density contrast to do its imaging. In our case, that wasn't an option. The fossil and the surrounding limestone matrix were almost uniform in density. However, there was a great distinction in colors. So fossils are red and limestone is white. And what you're looking at here, once it advances, is a movie which is taking you through successive layers which have been ground of a specimen. And essentially, the routine for automating the tracing of one particular specimen is to take a small section, then isolate a certain set of range of values for pixels of a certain hue, color, and intensity, and then run an algorithm to trace that specimen. Those layers are then positioned back in space at the same interval at which they were ground, which in this case is 50 microns, or approximately the width of a human hair. And then you get lines, which are then turned into points. The points then become meshes. And eventually you get a digital model, which can then be turned over to paleontologists who can interrogate it for morphogenetic qualities, as well as run a battery of tests, quantitative tests for things like porosity and permeability.
So after this process, what we're left with is an object about the size of your thumbnail, asymmetric in form, with a stalk or a holdfast and an interior network of water canals. These are diagnostic properties of a sponge, Earth's most primitive animal. And it turns out this finding moved back the fossil record of animals by more than 90 million years to beneath the Ice Age, suggesting that some animal lineage must have survived the Snowball Earth. Now, the one problem with this study was that it was prohibitively time-consuming. We needed to find a way to automate and make more precise the process of serial imaging and grinding and imaging.
So this is the new instrument. And what you have essentially is a precision CNC grinder for which we've designed a retrofit housing and imaging station. So we begin by grinding a predetermined depth from the sample, and then the specimen moves from the grinding position to an imaging position where a camera is actuated. So throughout the entire process, your entire sample is destroyed, and a digital archive is established, which documents each successive layer. This instrument is going to be built thanks to funding from the National Science Foundation, and it's going to become the center of a laboratory for digital fossil reconstruction at Princeton University.
So while the lab will continue to reassemble a host of paleontological samples, we're starting to explore other problems and other challenges in other fields. So as just one example, this is a specimen that was given to us by a geoengineer studying carbon sequestration. She wants to know, if you take CO2 out of our atmosphere that we continue to put in from fossil fuel burning and try to sequester it deep in the earth and rock, where does it go? And using our techniques, we can image in 3 dimensions exactly where that carbon dioxide goes, how it reacts, and whether this is actually permanent storage. So in essence, our lab is going to become a place for virtually any discipline to try to extract, reconstruct, and analyze 3-dimensional objects buried in a variety of media. So thank you.